So you can see in digestion, you can know, you know this, your pancreatic juices has enzymes and your intestine has enzymes. You know this process. HCl will digest here. It will digest HCl because it is acid. You can see folded protein will be denatured. This folded protein will be unfolded here in this stomach by acid. Acid causes denaturation, not denatured protein. What is denaturation? There will be alpha bond, there will be different type of bond, there will be different, this bond will be broken down by acid and it will be straightened so that the enzyme will act here. So now when a straight protein, denatured protein reaches this stomach, after this stomach is actually denatured, and some this pepsin act here also, it will digest, it will not digest completely, it will digest into peptones, small peptide, like this. Now these peptones, many of the peptones and some proteins will reach this intestine. Here intestine will have a very powerful enzymes from pancreas. These powerful enzymes, there are many powerful enzymes. These are trypsin, chymotrypsin, and then this carboxypeptidase. So this is tea cake. You can remember CEC, tea cake. So this tea cake enzymes will be secreted here, and these enzymes will digest this. And on the intestine, on the enterocyte, because intestine is I or E. So IE here you can see is a. So this is a vowel relate to this. So that is why I have remembered this aminopeptidase the way, because A is vowel and enterocytes are vowel, intestine is also vowel. So that is how I related that. And I memorized this aminopeptidase is present during my UG. I remember that way. Okay, so you can use that. Aminopeptidase is present in enterocyte. And now rest of the protein, because protein digestion is one of the difficult process for our body. So protein completely digest, it is not completely digested. If you take too much of protein, it is not digested. What happens is the protein that goes into the colon, and protein in colon that will be acted upon by bacteria, that becomes food for bacteria. So actually generally remember as a term. So we take daily 100 gram of protein. Average, it is not average. Actually 1 gram per kg of body weight. The normal actually calculation, 1 gram per kg of body weight we need to take every day, the protein. So I have, this is a round figure of 100 gram, because round figure is memorized by brain easily. So 100 gram is taken, 100 gram of protein easily excrete of our body, from our body. Actually it is lost every day by catabolism, other processes. 100 gram of protein is lost every day, and 100 gram protein is also there in your blood pool. This is a pool, amino acid pool. So 100 is a very famous number, you can remember. That is why 100 is here. So you can remember that. Now, what will happen? First, in gut, you know, in stomach, pepsin is stimulated by H+. So we will focus more about enterocyte, intestine. So in intestine, there is one enzyme, enterokinase. This enzyme is particularly purposed for activation of this trypsinogen. So trypsinogen will be activated first in intestine. After it is being secreted from pancreas, it is activated by enterokinase. Now trypsin is the master enzyme which will activate other enzyme, and its own activation is done by trypsin. And you know how is it? It is activated by proteolysis. First is this, these are the proenzymes. All are secreted from pancreas in the form of proenzymes. Why? There is significance. So because this enzyme should not be active in pancreas, should not harm pancreas. That is why the proenzyme is secreted and in the intestine, and it will be activated in the region where it is required. So it is required in lumen of intestine, and in lumen of intestine only it is activated this way. First enzyme activated is trypsin by enterokinase, that is present on the brush border of this intestine, and this trypsin will activate all tea cake. It will activate tea cake. What is tea cake? Tea is trypsin, its own activation, trypsinogen, and chymotrypsin and elastase, and then other is carboxypeptidase. Tea cake will be activated here. You can see here. There are two types of carboxypeptidase, A and B. Just remember for the time being. Now you can see the whole process of this digestion. So this is one picture, but very, very important, very informative picture. You can see the pepsinogen is activated by H plus. This is proenzyme. All digestive enzyme is secreted in the form of proenzyme. So pepsin will be converted into pepsin by HCl. And this pepsin actually can convert its own, it will cause its own activation later, the same way trypsin does. And then pepsin will act on proteins, long peptides, and it will convert into peptones, these small peptide, polypeptides. These polypeptides, and actually not all protein will be acted. Some protein again will also be there. Peptide will be there. Peptones plus polypeptide, big polypeptide proteins, they will reach again intestine, small intestine, duodenum. In duodenum, what happens in second part of duodenum? You know, the second part of duodenum, pancreatic juice is secreted. In that, there what will happen? This protein is acted upon by this all enzymes, and you already know the activation of this enzyme. How is it enzymes? First, trypsin is the first thing to activate, and then this trypsin is the master. Okay, this tree is the master. Okay, you can remember tree. Tree is the master, and tree will activate all this. So trypsin will activate this TCAC. Remember TCAC. What is TCAC? Trypsin, its own activation. Trypsin will activate. CAC is chymotrypsin. E for elastase, and then this carboxypeptidase AN. This, after it's being activated, so it will divide this protein into furthermore small peptide, and then after it's been done, digested, what will happen? The other further this dipeptide and very small peptones will be digested again by this brush border enzymes. This brush border enzymes are, one is, I told you, aminopeptidases. They are present here in intestinal epithelium. Aminopeptidases are present in intestinal epithelium, brush border of intestine. You can see here 4 types of aminopeptidases. WPAN, you can remember WPAN. So even if you don't remember, that is not a problem, but I have given here so that it will be more information for you. Different type of aminopeptidases are there. So I told you this, there are 2 types of, if we classify this all peptidases, whole, this brush border plus this all, you can classify this all peptidases into 2 types, endopeptidase and exopeptidase. Exopeptidase. You know, endopeptidase means, I'll tell you what is endopeptidase. Endopeptidase will act here. Endopeptidase will act here, inside, here, inside. But exopeptidase will act here from the outside, either from carboxy terminal or from amino terminal. If it acts from amino terminal, it is aminopeptidase. You can tell the name came derived from this because it acts from amino terminal. If it acts on carboxy terminal, you can call it carboxypeptidase. Carboxypeptidase. And you know this, as a matter of fact, you can see this aminopeptidase and carboxypeptidase has different types, not this trypsin and chymotrypsin. Aminopeptidases are of 4 types. Carboxypeptidases are of 4 types because nature knew from the beginning that at the end of each peptide there will be different amino acids. That is why in order to act on different amino acids, there will be different. So you can see W is designation here. W comes. W is for tryptophan. 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 So I have not written trip. Tryptophan. The pronunciation is tryptophan. So that is what this W came with. This tryptophan resembles W. And if tryptophan is here, so it will digest tryptophan. P is for proline. P is for proline. So if proline is here, this P will digest. A for amino, anionic amino acid. What are anionic? Glutamate. Glutamate is E, and aspartate D. So D and E, these are the amino, not like anionic, anionic. These are anionic amino acids. This anionic amino acid will be acted upon by this. This N for neutral. Neutral. What are neutral amino acids? Many neutral amino acids are there. Many neutral. Glycine. You can see valine, leucine, isoleucine, like sulfur-containing amino acid cysteine. Then your methionine. These all amino acids are neutral. So this neutral aminopeptidase will act here. If neutral amino acid is present here, it will act. So that is why, that is the significance of presence of these 4 types of aminopeptidases. But in exam, if you may not find, but understanding, if you understand, you will have a sense of satisfaction. So that is the reason I have actually presented here. This is a very good picture I got from somewhere, and then I put here. I got from this elsewhere.com. So that is why I put here, and this is very like trusted picture. So you can trust on this picture. This is from elsewhere. You can see now, this don't focus on this carboxypeptidase P. Okay, it is exon proline. Don't focus on this carboxy. Just remember carboxypeptidase A and B. They are pancreatic enzymes. A will act on A for, just remember, A for aromatic, and B for basic. Though it's not the name, but B for basic, A for aromatic resembles basic. So it will act on the amino acid. If this amino acid is aromatic, then this carboxy A will act. If this is at the amino group, it is basic amino acid, B will act. Other amino acid, this P, carboxypeptidase P will act. But this is not that important. Carboxypeptidase P is not that important. Remember carboxypeptidase A and B. And this is the different type of carboxypeptidase. I have already told, this is zinc containing metalloproteases. All are serine proteases. So understanding of this proteases will actually make you understand whole thing. Now after protein being digested, what will happen? There are many transporters, not only single types of transporter. We have seen in glucose transporter, glucose transportation is very simple. There is only one transporter, as GLUT is there in intestine, which is secondary active transporter. These are also, many of them are secondary active transporters similar to this, but there are other different type of transporter also. Predominantly they are transporters similar to S-GLUT. Okay, you can see here this transporter, these are symporter. Many of them are symporter. You can see this symporter. Okay, many of them are symporter here. Symporter, you can see symporter. Many of them are symporter. Some are antiporter as well. So predominantly similar to S-GLUT. So you can use that concept here also, S-GLUT, secondary active transfer. Now, so this is whole thing about the most important slide about the digestion. If you remember this slide, so every digestion is already done. Digestion and absorption is already done. And you can see here ACE protein, angiotensin converting enzyme protein. You can see this is also peptidase. That is why I have written here. And this is actually very important for us, because you know this coronavirus, our COVID-19, not our COVID-19, our enemy. This is the enemy of humanity right now. So COVID-19 will bind this ACE inhibitor, ACE. That is why it is said that ACE inhibitor will act. So this is an extra information, concurrent information for you. So you can remember this ACE is present in brush border of intestine and brush border of your lungs, actually respiratory system. ACE is an peptidase. ACE is an what? ACE2 is an proteases, serine proteases. Now you can see here this protein digestion. Protein digestion, as I've told you, endopeptidase will act inside here. So you already know, I have already told you endopeptidase. What is endopeptidase? This T, C, H, E. These are except last C. And if you remember one A, it will become attach. So attack, you can call it attack. So attack is endopeptidases. And what is exopeptidase? Last C, carboxypeptidase. Sorry, this A is not there, so I missed this. But ACE is there. ACE is there. So TAC is your endopeptidase, and A and C, aminopeptidase and carboxypeptidase, are exopeptidase. So remember these two examples, exopeptidase. Amino because from amino terminal, carboxy because it will act from carboxy. So you know this. You can understand this easily. Okay, exopeptidase further breaks oligosaccharide produced by endopeptidase. So endopeptidase act, and then it will act on this. So aminopeptidase, amino terminal, it is present on breast border, and carboxypeptidase, pancreatic enzymes. This is how it will happen. We have already done this. So this will, the whole protein like this, it is like this. So it will digest this folded protein, it will unfold because of acid, the protein will be unfolded and then digested into polypeptides. And polypeptides will be acted upon by this trypsin and other enzymes. PK. Okay, PK. It will be acting TK and then it will digest dipeptide, tetrapeptide, this type of peptide. Now, irripsin is intestinal. All intestinal proteases are commonly called irripsin. Okay, small intestinal brush border are called irripsin. Irripsin is a mixture of enzyme containing protein fraction found in intestinal. So these all irripsin. You got this irripsin. So these irripsins will digest this further. Carboxypeptidase and this digest. Now, so you can see the whole digestion process food will be masticated. It will be little bit mastication, denaturation will happen. Then it will reach gastric mucosa in stomach. It will be digested more. Again, it will reach small intestine. Its small intestine will be digested. And then the left out will reach your this large intestine. Large intestine, it will become a food for your bacteria, your good bacteria. There are many good bacteria also are there, good bacteria, bad bacteria also. So if you take in limited amount, it will actually nourish good bacteria as well. So along with the salads, if you took salad and then green vegetables, these green vegetables are very good food for your good bacteria. Saprophytic, your helping bacteria. That is why previously people used to say, we used to talk about nutrition as this: protein, carbohydrate, and mineral. Protein, macronutrients, protein, carbohydrate, lipid, and micronutrients, vitamins and minerals. But see, if you only take this, you will not survive. So in recent days, we need to consider water as also nutrition. We forget water. Water is also a nutrition. Because water is abundant, we don't consider as nutrition. It is nutrition. Water is nutrition. In addition to water, this salads, green vegetables, these are nutrition because these are food for your normal bacteria. If you feed this normal bacteria, this normal bacteria also help you making thin. Okay, because this normal bacteria is contributing for your immunity. Normal bacteria is contributing to fight your cancer. So normal bacteria, the gut bacteria, has very, very important role. It is very big. It is like God within you. Many tiny God within you, helping you to survive. So you need to provide food for them also. But we don't think. We are very selfish people, either because of lack of knowledge, or even we have knowledge, we become selfish like that only. So we need to take care of those bacteria, those saprophytic bacteria, by these green vegetables, and then few proteins. Okay, proteins, left out proteins, and few like green vegetables, salads. These are very important food for these bacteria. You need to take daily, and that bacteria is very helpful for you, fighting infection, everything. So this left out food will be food for your bacteria, and that is why this bacteria will break, and this is actually the flattest part. Okay, that actually form your flatus. Ammonia, okay, smell like a bad smell of this flatus is because of this nitrogen breakdown, this amino acid breakdown. So these are the bacteria you can go through. Anaerobic bacteria, all anaerobic bacteria form this. Okay, most of them are helpful, and you can so this is one clinically very important. Low protein diet, laxative and sometimes antibiotic used to limit protein fermentation in disease. If a person is in disease condition, so you have to limit this fermentation because there is production of ammonia. So you need to limit the production of ammonia by these processes. So these are the proteins I have already told you. So you can see ELA, elastase is ELA. So elastase, aliphatic. So it will act on aliphatic. These are endopeptidases, and it will act on a region where is aliphatic. Aliphatic means straight chain. A straight chain amino acid is aliphatic. Now, for example, glycine, alanine, your branch chain amino acid, valine, leucine, these are all aliphatic amino acid. Now, chymotrypsin will act on your this aromatic. Okay, I actually make this O very big. Chyme O trypsin. So O is aromatic. This is how I remembered in the beginning. I don't need now, but in the beginning I remembered this way. Chyme O, made this O very big and remembered this. So you can use that. Chymotrypsin act on aromatic amino acid, especially, and then neutral amino acid, chymotrypsin, and then this carboxypeptidase and A for, as I've told you, aromatic, B for basic, arginine, lysine. You can easily understand this. And tree. So how did I remember trypsin? So I remember trypsin as like tree. Okay. So tree always has a base. So it will act on basic amino. This is how I remember. So you can use that. Trypsin will act on basic amino acid. Wherever basic amino acid located inside the it will act here. So you can easily remember them through my trick, and you can develop your own trick. But trick is important to remember these complicated things, seemingly complicated, not complicated. What would happen to pancreatic proteases after they digest all the protein? So what will happen? These pancreatic proteases all will be digest themselves, because remainder of pancreatic proteases will be dangerous for your intestine. So they will digest themselves and again will be absorbed in the form